Hello, and welcome to the Research Continuing Education series for licensed massage therapists, brought to you by the Massage Scientist. In this presentation, we will explore a recent research paper in the field of massage therapy, going into depth on key terms and ideas, and explaining the methods, results, and conclusions of the publication. The intended audience for this presentation is licensed massage therapists practicing in Washington State, but any massage therapist, physical therapist, or other healthcare worker can find interesting and useful information for their practice as well. We'll begin with an overview of the presentation, then proceed to discuss the specifics of the research paper with particular focus on making its motivations, methods, and results easily understandable to massage therapists who do not happen to have an advanced scientific training. The entire presentation will take about 1 hour. At the end of the presentation, you'll find information on how to receive a continuing education certificate, provide feedback to the instructor, and follow up with any questions. Let's get started. Let's begin with an overview of this presentation. We will start with a brief discussion of why it's worthwhile to study massage therapy research, with specific focus on the value of online continuing education as it applies to the field of massage therapy, and how such study can benefit the practicing massage therapist. I'll then have a few words about myself, my scientific and professional background, and my goals for this presentation. Following this, we will dive straight into the paper, dissecting the title for its core ideas, and examining the abstract of the paper to pick out the key terms, ideas, and techniques. Since those are the essential components for the research required to understanding its value and importance, we will spend some time discussing each of those key terms and techniques to ensure understanding of how each one fits into the overall research. The methods for this paper are also fascinating, so we'll spend a few minutes looking at those as well. Following this, we'll look in detail at the results of the research, analyzing the data so that we can follow the logical conclusions of the scientists as they attempt to answer their hypothesis. Discussion and conclusions will follow, addressing the central hypothesis of the paper, assessing the methods used, and noting any unexpected results, while also identifying areas for further research. Having covered the research in detail, we will then briefly extend the discussion beyond the paper itself by talking about some of the implications of the research, with further questions that might be interesting to explore. The presentation concludes with information for massage therapists wishing to receive a certificate for this continuing education, along with links for feedback and to contact the instructor with any questions. So why study massage therapy research? Washington State requires licensed massage therapists to take a minimum of 24 hours of continuing education every 2 years, of which 16 hours may be online, that is, not hands-on. So why study online CEs? As an online course, this research CE presentation can be explored at almost any time or place, with a computer, or a tablet, or a phone, and at your own preferred pace. You can pause the presentation to refer to other sources, make notes to improve your learning, or save your progress and then resume study at a later time. You are in complete control of your experience, and I encourage you to think of yourself not just as someone watching a video online to fulfill a requirement, but as a student studying a topic for personal engagement and professional edification. That remains true whether you are watching this presentation during business hours in your office or studio, or in your bedroom at night eating a carton of ice cream. Online CEs are some of the most flexible, customizable hours of study possible. But why study research CEs in particular? Research is a wonderful area to fulfill your required CE hours. You'll be reminded of aspects of your foundational anatomy and physiology training. You'll expand your clinical reasoning and deductive thinking. You'll increase your mastery of the scientific language that underscores massage therapy as a health science. You'll develop confidence in your knowledge and familiarize yourself with some of the most current research happening in massage therapy today. Your clients will appreciate your professionalism and expertise all the more when you are able to answer their questions from a position of deep knowledge of your field. And you yourself may gain a new appreciation for massage therapy as a field you can be truly proud to be part of. It's an exciting time to be a massage therapist. Let's do a brief introduction, and then we'll dig into the meat of the research paper that we'll be discussing today. By way of introduction, hi, my name is Peter, and I'm a trained scientist as well as a practicing licensed massage therapist. My scientific background is in biochemistry, or the chemistry of living things, and I have degrees from Western Washington University in Bellingham, Washington, as well as the University of Oregon in Eugene, Oregon. Beyond this, I have over a decade of experience in academic instruction and biotech industry research. I am co-inventor on 2 U.S. patents, one for research tools and one for methodologies, and have presented research at academic and industrial conferences around the U.S., including the presentation of original massage therapy research at the Massage Therapy Research Conference in 2016. As a licensed massage therapist, I have practiced continually since my licensing in 2015, with experience in massage therapy clinics, chiropractic clinics, corporate offices, and on-call mobile massage services. I'm currently self-employed as an LMT in the greater Seattle area. My goal for the LMT Research Continuing Education series is to use my knowledge and background in science to communicate to my massage therapy colleagues the exciting potential of current research in massage therapy as a tool not just for improving their own confidence and professional acumen as scientific healthcare providers, but also as a platform where they can learn about actionable, evidence-based research that can inform and empower their own practices. At the end of the day, we do what we do in the service of improving our clients' health, and I want to keep that uppermost in mind as we talk about some of the latest research in massage therapy. Let's get started. So let's get right into it. Today's research comes to us from colleagues at the Department of Rehabilitation Sciences, Center for Muscle Biology, University of Kentucky in Lexington, Kentucky. Corresponding author, Timothy A. Butterfield. A few words about the journal. Physiological Reports is a peer-reviewed, open-access, online-only scientific journal covering original research in all areas of physiology. It is published by Wiley Blackwell on behalf of the Physiological Society and the American Physiological Society. This journal was established in 2013, meaning that it's far from old and stodgy. It's also important to note that this is a peer-reviewed journal, which means other scientists who were not part of the study but who are considered experts in the same field reviewed the work before it could be published. Peer review is an important test for any research, and you should look for that in any journals you use to follow current research. It is also important that this journal is open access, meaning the full text is freely available without a subscription. Too much publicly funded research is hidden behind a paywall, where few LMTs can access it. In this series, we are committed to highlighting open access research so everyone can enjoy the benefits of massage therapy research in their professional practice. A direct link to this publication is also posted in the description for this presentation. I strongly urge you to bring up a copy on your computer to refer to, or better still, print out a paper copy so that you can refer to it or even make notes on it during the presentation. This will improve your learning and retention of the material. Let's discuss the title first. I know some of you are already glazing over as you look at unfamiliar words, but don't despair. As we go along, I'll break everything down for you and spend quite some time expanding on the key terms and ideas so that you'll understand at every point what the research is about, why it's important, and what it means. Let's start by talking about the title phrase by phrase. Massage increases satellite cell number. That tells us the technique being examined, that is, massage, and what the primary effect of that technique the research is focused on, the increase in satellite cells. For the moment, we don't need to worry about what those are or what they do, only that the technique, massage, causes their number to go up. On to the next phrase. Independent of age-associated alterations. That means the same result is observed in both young and old subjects, which means we'll be comparing those two groups. In sarcolemma permeability. Permeability is the capacity for permeation, or how easy it is to make holes in something such that material can enter or exit. That something is the sarcolemma, which should be familiar from your anatomy and physiology classes. It's the cell membrane of a striated muscle fiber cell. So take that all together, and we now know that this paper is talking about how massage increases the number of satellite cells independent of the integrity of the cell membrane of skeletal muscle cells, whether the subject is young or old. Since massage is all about affecting muscles, it's great that we have a clearer sense of what the authors want us to know about their findings. Let's expand on that with the next section of the paper, the abstract. Let's dig into the abstract. This is where the authors summarize the key points of their research: the background of the work, the motivation for their research, the question or hypothesis they wanted to test, the materials and methods used for their research, and the results of their work. Let's take each section in turn, and as you follow along, take note of the key terms and ideas I have highlighted in red or bold text. We'll be going over these in much greater detail shortly, but for now, just try to follow the overall narrative of this summary. Abstract. Massage is a widely accepted manual therapy used to modulate the inflammatory response of muscle and restore function, but prolonged compression of muscle potentially causes overt injury and damage to muscle fibers. Therefore, a balance exists between the positive effects of massage and the induction of mechanical damage and injury. In addition, skeletal muscle of aged individuals displays increased stiffness, and therefore the response to massage is likely different compared with young. We hypothesized that the aged skeletal muscle exhibits increased sarcolemmal permeability when subjected to massage compared to young skeletal muscle. Male brown Norway F344 rats, 10 and 30 months of age, were each divided into control, non-massaged, n equals 8, and massaged, n equals 8, groups. The right gastrocnemius muscle received one bout of cyclic compressive loading for 30 minutes at 4.5 newtons as a massage mimetic. Muscles were dissected and frozen 24 hours after massage. Alterations in sarcolemma permeability were quantified by measuring the level of intracellular IgG within the muscle fibers. Immunohistochemistry was performed to determine IgG inside fibers and Pax7+ cell number as an indicator of stem cell abundance. Average IgG intensity was not different between control and massaged animals at either age. However, a significant shift to the right of the density histogram indicated that massaged animals had more fibers with higher IgG intensity than control at 10 months. In addition, Pax7+ cell number was significantly elevated in massaged muscles compared with control at both ages. One bout of massage did not induce overt muscle injury, but facilitated membrane permeability, which was associated with an increase in satellite cell number. Data suggest that the load applied here, which was previously shown to induce immunomodulatory changes, does not induce overt muscle injury in young and old muscles, but may result in muscle remodeling. Okay, there were a lot of scientific terms and ideas there, and the condensed form of the abstract might make it a little hard to follow the logical narrative of the research study. Let's go into detail on several terms here so that we can get a better idea of what this research paper is all about. Then we'll put all the information together to understand the flow of the research. I understand that abstract might be a little dense and hard to follow, so I've pulled each of the key terms that I want to talk about out of the abstract and placed them here in a bullet point format. We'll talk briefly about each of them, and those that are more scientific or technical we'll dig into in detail in a moment, so that you understand how each of them plays into the logical reasoning of the research paper as we go forward. Massage is a manual therapy widely used to treat a variety of musculoskeletal conditions, and may be defined as the manipulation of body tissues with rhythmic pressure and stroking for the promotion of health and well-being. Skeletal muscle is a highly voluntary, responsive tissue that permits movement of the skeleton and alters readily to adapt to a variety of physiological and pathological stimuli. This adaptability is due, at least partly, to the constant turnover or remodeling of muscle proteins, membranes, and cell populations. Sarcolemma is the cell membrane of striated muscle fiber cells. Permeability is the degree to which fluids and cellular materials can freely enter or exit the cell, and is a measure of normal homeostasis or injury. Norway rats, F344. Their color is albino, and this strain originated in 1920 with two researchers, Curtis and Dunning, from Columbia University Institute. The breed was transferred to the National Institutes of Health in 1951. This is easily the most widely used general-purpose inbred rat strain, being particularly favored for cancer research and toxicology. Their typical lifespan is 30 months. Now that's important because we're using old rats at 30 months. Control groups are identical in all characteristics except for the intervention technique, in this case, massage. N number is the number of individuals tested within a control group. Important to note for statistical purposes, the higher the N, the greater your confidence that any changes observed are the result of the intervention rather than random chance. 4.5 newtons. A newton is a unit of force, analogous to pounds. 4.5 newtons is just over 1 pound of force. Intracellular IgG. Immunoglobulins are antibodies, the basic immune protein involved in the recognition of foreign matter and the signal for an immune response to begin. Healthy sarcolemma is impermeable to large extracellular proteins. Therefore, the presence of IgG inside the sarcoplasm of muscle fibers is indicative of greatly increased muscle permeability. Immunohistochemistry, also known as immunostaining, is a technique of visualizing tissues and cellular structures using immune proteins labeled with a molecule visible to a microscope. PAX7+. Adult skeletal muscle has a remarkable regenerative capacity due to a rare population of cells, termed satellite cells, that are wedged between the basal lamina and the sarcolemma, interspersed along each adult muscle fiber. PAX7 itself is a gene that encodes a transcription factor, the expression of which generates new muscle cells to aid in repair and replacement of damaged muscle cells. A histogram is an approximate representation of the distribution of numerical or categorical data, usually represented by frequency bars under a curve. The classic example of a histogram is the bell curve. Immunomodulatory means anything that affects the function of the immune system, generally either to stimulate it or to dampen it down. Satellite cells are the resident stem cells of muscles. They reside between the basal lamina and the cell membrane of muscle cells or fibers. Satellite cells remain mostly quiescent in adult muscle, but become activated during insult to the muscle that is most often recognized as muscle injury. Once activated, satellite cells divide and aid in the recovery of the injured muscle cells to repair damaged muscle. And last, muscle remodeling is the active process by which cells are repaired, replaced, or added to. Alright, that was a lot of words. Let's bring it back to something that I know you know well: skeletal muscle. Skeletal muscle is one of three major muscle types, the others being cardiac muscle, found only in the heart, and smooth muscle, generally associated with internal organs. Skeletal muscle is a form of striated muscle tissue and is under the voluntary control of the somatic nervous system. Most skeletal muscles are attached to bones by bundles of collagen fibers known as tendons. This is the major muscle type that we, as licensed massage therapists, are concerned with. In the case of the research being presented here, the key muscle we are concerned with is the gastrocnemius of the test animals. Remember from the abstract, the right gastrocnemius muscle received one bout of cyclic compressive loading for 30 minutes at 4.5 newtons as a massage mimetic. This is important to note because while we as LMTs are aware of contralateral effects, each animal provides an internal control of one gastrocnemius that received massage and one that did not. In this way, this provides a means of testing contralateral effects as well as massage or no massage control groups. More on that in a moment. The sarcolemma is one of the key structures examined in today's paper, so let's go into a little bit more detail on what it is and how it works. The word sarcolemma comes from two Greek words, sarx, meaning flesh, and lemma, meaning sheath. It's the cell membrane of a striated muscle fiber cell. It consists of a lipid bilayer and a thin outer coat of polysaccharide material, or glycocalyx, that contacts the basement membrane. The basement membrane contains numerous collagen fibrils and specialized proteins such as laminin that provide a scaffold to which the muscle fiber can adhere. Through transmembrane proteins in the plasma membrane, the actin skeleton inside the cell is connected to the basement membrane and to the cell's exterior. At each end of the muscle fiber, the surface layer of the sarcolemma fuses with a tendon fiber, and the tendon fibers in turn collect into bundles to form the muscle tendons that adhere to bones. The sarcolemma generally maintains the same function in muscle cells as the plasma membrane does in other eukaryotic cells. It acts as a barrier between the extracellular and intracellular compartments, defining the individual muscle fiber from its surroundings. The lipid nature of the membrane allows it to separate the fluids of the intra and extracellular compartments, since it only selectively permeates with water. Now, permeability is the degree to which fluids and cellular materials can freely enter or exit the cell, and it's a measure of homeostasis or injury. Permeability is essential for delivering nutrients, including oxygen and glucose, while also removing waste products such as carbon dioxide. At the same time, intracellular and extracellular materials and structures must not pass through this membrane, and those include the cytoplasm, organelles, DNA, and so forth. When injury causes poration of the sarcolemma, sometimes called microtearing in exercise physiology, these internal materials can escape the cell and create an immune response that gives rise to inflammation in the surrounding tissues. This inflammation is a signal for various repair mechanisms, one of which we'll examine in just a moment. Well, I guess we're the control group. The control group is defined as the group in an experiment or study that does not receive treatment by the researchers and is then used as a benchmark to measure how the other tested subjects respond. The other groups are collectively referred to as experimental groups and receive some form of intervention, but are otherwise identical in character to the control group. This keeps as many variables as possible fixed, so that the only variable that changes is the one being tested. In this study, there are two kinds of control groups. First, the set of animals which did not receive massage. Second, among those animals that did receive massage, only the right gastrocnemius was massaged, leaving the left gastrocnemius as an internal control. Now, the size of the control group and the experimental group also matters. The N number is the number of individuals tested within a control group. Important to note for statistical purposes, the higher the N, the greater your confidence that any changes are the result of the intervention and will remain true when applied to the world outside the study. This is why, in a lot of massage therapy research involving humans, the data is often of low statistical or predictive value because relatively few individuals are studied. In the cartoon shown here, the N number is 2, that is, 2 experimental mice, 2 control mice. In the research we're studying today, the N number is 8. Both the control groups and the experimental groups have 8 members. As we'll see in a moment, there are actually 4 groups being compared in today's paper, but each of them has 8 members. As a side note, anecdotal evidence is generally considered to be N equals 1, that is, 1 experimental subject and no control group. And for this reason, it's not considered in the same category as well-controlled experimental data. As massage therapists, we recognize its value to the client, however, and can even report such experiences with a case study report. Over time, case studies can form a body of evidence that supports a larger study with appropriate control groups and N numbers. Our next key term is IgG. The abstract of the paper mentions intracellular IgG. Quote, alterations in sarcolemma permeability were quantified by measuring the level of intracellular IgG within the muscle fibers. End quote. IgG is an abbreviation of the word immunoglobulin. Immunoglobulins are antibodies, the basic immune protein involved in the physical recognition of foreign matter and the signal for an immune response to begin. Individual antibodies recognize an incredibly specific molecular shape. Upon binding it, they signal to other immune proteins and cells to attack, engulf, and degrade the foreign material that they've bound to. The structure of IgG, above, shows two antigen binding sites up at the top. This is where the antibody binds to its target. The Fc region at the bottom of the image is a highly conserved sequence, the same in almost every one of your antibodies. That makes it incredibly useful for a scientific detection technique called immunohistochemistry, which we'll examine in a moment. In order to understand immunohistochemistry, however, you must know about the structure of IgG. Now, while the figure at left is a useful symbolic representation of an IgG, take a look at the more accurate space-filling model shown to the right. You can easily see why scientists first called it a globulin, but also notice how detailed and intricate the antigen binding domains are up at the top of the molecule. This is what gives them their exquisite specificity for a unique target based on its three-dimensional molecular shape. The important point for the paper we're discussing is that as a lipid bilayer membrane surrounding a muscle cell, healthy sarcolemma is not permeable to large extracellular proteins like IgG. Therefore, if the researchers find evidence of IgG inside the sarcoplasm of muscle fibers, it will indicate increased membrane permeability and perhaps poration or a microtearing injury. So how do the researchers tell whether there is IgG inside the muscle cell? Let's talk about immunohistochemistry. Immunohistochemistry, or IHC, is a technique for visualizing the presence of specific subcellular structures using immune proteins, especially IgG, labeled with special fluorescent, radioisotopic, or enzymatic reporters. First, look at the model of the IgG in the upper left corner. It should be familiar from the last slide, just flipped upside down. The lower IgG, labeled here as the primary antibody, is binding to its antigen target, that is, to the unique protein molecular structure that it recognizes, and the point of recognition is at its antigen binding site. But at the opposite end of the primary antibody is the highly conserved Fc region. Using biotechnology, scientists discovered it was possible to make antibodies that specifically recognized the conserved Fc region of other species' antibodies. So the secondary antibody in this case might be an antibody from a goat that binds to the Fc region of the rat antibody. This is useful because if our model animal is a rat, as it is in this case, and it is the rat IgG that has potentially entered the muscle cell through a permeated sarcolemma, our goat anti-rat IgG antibody should bind to it and nothing else. That gives researchers a clean, clear, unambiguous picture of what is happening inside the cells under investigation. Now, notice the protein attached at the very top of the secondary antibody in yellow. This could be any number of reporter molecules, including the enzyme HRP, horseradish peroxidase, as in today's paper, which is a fluorescent molecule. This is what we were actually seeing when we examined tissues with IHC. In the model shown here, we have a two-step IHC process. First, the primary antibody is exposed to thinly sliced tissues and allowed to bind. Then any unbound material is washed off, leaving bound primary antibody behind. Second, an antibody that recognizes just the conserved Fc region of the primary antibody is introduced, followed by a second wash. The second antibody contains an attached enzyme, HRP, which can make a small reporter molecule produce fluorescent light under a fluorescence microscope. What we are looking at in the slides labeled A through C above are tissue sections, that means extremely thin slices of muscle, from the rat gastrocnemius described in the research paper we're studying. You can see how in pictures B and C, the fluorescence is found only outside the muscle cells, whereas in image A, it is also within the cells. Look for those arrows. This shows that IgG has gone from the extracellular space to the inside or intracellular space of the cell. These images were generated with a fluorescent microscope. Using a device called a fluorimeter, the researchers can measure the actual amount of fluorescent light emitted by these cells and make a graph of it. And these graphs will allow us to quantify and compare the fluorescence of different control groups, as we'll see in just a moment. Our next key term is density histogram. Similar to a bar graph, a histogram is a distribution of numerical or categorical data that permits numerical trends to be observed visually. The classic example is a bell curve, where a statistical distribution of frequency is patterned smoothly around the mean. But in the real world of experimental research, data is rarely as clean and symmetrical as a classic bell curve. Here, we are looking at histograms showing data from our research paper. On the vertical axis, we have number of fibers from the tissue sections in the previous IHC slide. And on the horizontal axis, fluorescence intensity, measured from our fluorescence microscopes using a fluorimeter. The four histograms correspond directly to our two control groups and our two experimental groups. What we see here is that the researchers counted the number of fibers present in each sample type that had the same amount or density of fluorescence intensity and grouped them together in a single bar, allowing them to visualize the distribution of populations of different fluorescence intensity throughout the sample. But what does it mean? Now, if you remember, the abstract of the paper said, quote, alterations in sarcolemma permeability were quantified by measuring the level of intracellular IgG within the muscle fibers, unquote. And later, it went on to say, quote, a significant shift to the right of the density histogram indicated that massaged animals had more fibers with higher IgG intensity than control at 10 months of age, end quote. The shift to the right in the density histogram is shown in figure B of the data above. You can see that it is the only histogram with two curves. That is the shift referred to in the paper. This means that out of all four groups examined, only young, massaged animals demonstrated increased sarcolemma permeability. This was a surprising finding to the researchers who had hypothesized that the older animals, with greater cytoskeletal restriction than their younger counterparts, would show greater sarcolemmal permeability and poration following massage. Instead, it was the younger animals. To help explain why this might be, let's examine the last of our key terms, satellite cells. We've now reached the last of our key terms for this paper, but there's several that are rolled into one, so pay close attention. Satellite cells are the resident stem cells of muscle. They reside between the basal lamina and the cell membrane, that is, the sarcolemma, of muscle cells or fibers. Satellite cells remain mostly quiescent in adult muscle, but become activated during insult to the muscle. Most often, this is categorized as muscle injury. Once activated, satellite cells divide, differentiate, and aid in the recovery of the injured muscle cells to repair the damaged muscle. Now, remodeling is an active process by which muscle cells are repaired, replaced, or added to during homeostasis. Skeletal muscle is highly responsive tissue that alters readily to adapt to a variety of physiological and pathological stimuli. This adaptability is due, at least in part, to the constant turnover or remodeling of muscle proteins, organelles, and cell populations through the action of satellite cells. Now, in the current study, researchers used immunohistochemistry, or IHC, to follow the activation of satellite cells as a means to visualize muscle injury and remodeling via satellite cells. How are satellite cells activated? Through PAX7. PAX7 is a gene found in some satellite cells that encodes a transcription factor. When this transcription factor protein binds to the satellite cell's DNA, it triggers the cell to generate new muscle cells to aid in repair and replacement of damaged muscle tissue. Scientists use the term positive to denote a cell that contains a particular gene, so they would say that some satellite cells are PAX7 positive if they contain that gene. In essence, PAX7 is the genetic switch that turns on a satellite cell, activating its role in promoting repair of injured muscle cells and the growth of new tissue. You can see in the image above how satellite cells are distributed along a skeletal muscle between the sarcolemma and the basal lamina. One satellite cell shown is Pax7 positive, and when an injury to the muscle cell is detected, probably through an immune cascade leading to localized inflammation, the Pax7 positive satellite cell divides and differentiates, growing new muscle cells to repair the damage to the tissue. This, in turn, attenuates the immune response, reducing the localized inflammation, and the Pax7 switch turns off, returning to a quiescent state. In other words, we can expect that following the mechanical pressure of massage as found in this study, Pax7 positive satellite cells might play a role in protecting muscle cells from microtearing, with the result that sarcolemma permeability might be prevented or rapidly repaired. Remember, for later, that it was young, massaged rats that showed increased sarcolemma permeability. We will want to know what their level of Pax7 plus satellite cells is when trying to understand this observation. Now that we've explored all the key terms and techniques for understanding this research, we can finally turn to the specific methods that the scientists used in their study. As we start talking about methods, it's important to remember our control groups and experimental groups that were used by the researchers in this study. We have 16 young rats aged 10 months and 16 old rats aged 30 months. And remember, 30 months is about the full life expectancy of Norway rats. Each of those two age groups is divided into a massaged group and a control or non-massaged group. This gives us 4 test groups as shown above and allows the researchers to independently compare both massage and control at a single age group, moving horizontally across this chart, or between 2 age groups who both received massage. That would be the first vertical column. When we talk about these groups, when we discuss the findings of this study, we will use the researchers' naming system. 10M and 30M mean the 10-month-old and 30-month-old massage groups, while 10C and 30C are the 10-month-old and 30-month-old non-massaged or control groups. Now, if you're like me, then you've probably been wondering this entire time, how exactly do you massage a rat? Let's look at the apparatus the researchers used. Let's talk about how you massage a rat. Now, while the paper that we're talking about today does not show the diagram shown in this slide, it cites previous work by some of the same researchers. I used those citations to find the original design of the massage device, and it's shown here. Briefly, rats were placed on a mesh sling in the left lateral recumbent position, that is, laying on their left sides, with the right hind limb secured to a small platform by self-adherent tape encircling the talocrural or ankle joint and midfoot. This placed the lateral aspect of the right gastrocnemius muscle facing superiorly for the application of a massage mimetic by a custom fabricated device. The cyclic compressive loading device is a spring-loaded mechanism containing a cylindrical roller that will glide over the muscle mass. The roller will be displaced in response to the normal force being exerted back on the roller from the muscle, and the force is measured in real time. For massage application, the roller is placed on the skin overlying the right gastrocnemius muscle immediately posterior and proximal to the lateral malleolus, and cycled proximal to distal and back along the length of the gastrocnemius muscle, applying 4.5 newtons, or about 1 pound of pressure, with a duty cycle of 0.25, that is, muscle compressions were performed at a frequency of 2 per second, for 30 minutes. So in essence, what the researchers used was a robot-driven foam roller controlled by a computer that applied the same amount of pressure to the muscle at all times in a tightly controlled path and frequency. Now, obviously this is not what most LMTs would recognize as massage, but the perfect robotic consistency of this device helped the researchers define a specific mechanical intervention, reducing human variability and allowing comparison between not only many different test subjects, but also between different studies and papers. As an aside, this is one of the key challenges in any massage therapy research. so it's useful to see one way that researchers have attempted to solve it. So, what happened to the muscle cells of the rats that were massaged with this robotic foam roller? Let's take a look. Now here's some things you can't do with your massage clients. After completion of the massage treatment or control sham treatment to the right gastrocnemius, rats were placed back into their cages and allowed to recover. 24 hours following the massage treatment, the animals were euthanized. Right gastrocnemius muscles were harvested, flash frozen in liquid nitrogen, and stored at -80 degrees Celsius for further analyses. Now that the researchers had tissue samples from young and old rats who had either been massaged or not, it was time to use immunohistochemistry to determine the effects of that massage. Alterations in sarcolemma permeability were quantified by measuring the level of intracellular IgG within the muscle fibers of the massaged and controlled gastrocnemius muscles of young and aged rats. Our healthy sarcolemma is impermeable to large extracellular proteins. Therefore, the presence of IgG in the sarcoplasm of muscle fibers is indicative of increased membrane permeability. Membrane permeability was determined based on the fluorescence density measurements inside the muscle fibers. A higher fluorescence intensity indicates a greater membrane permeability and greater infiltration of IgG into the muscle fiber. For injury-positive control tissues, IgG infiltration was measured in fibers from eccentrically exercised tibialis anterior muscle from a previous study, shown in slide A. So, from this positive control, you can see some of the cells are lit up with fluorescence. That indicates the presence of intracellular IgG, which in turn means the sarcolemma protecting those cells has been injured, allowing extracellular IgG to enter the muscle cell. So with this as a positive control, let's look at what the researchers found when they performed IHC on tissue samples from the 4 groups used in this study. Now, if you've stuck with me so far, congratulations, we've reached the payoff. It's time to talk about results. Now that we've gone into detail on the key terms and methods, we can dig into the results of the research study. Our first result is a fluorescence microscopy image from tissues taken from each of the 4 control groups, that's B through E above. These are immunohistochemistry samples, which means we are looking for fluorescence inside the cells to demonstrate extracellular IgG, which has entered the cell through sarcolemma permeations. As you can see from these 4 microscopy images, the overwhelming majority of the fluorescence lies between the cells in the extracellular space. That would indicate little or no sarcolemma permeability in any of the 4 groups tested, young and old, massage and no massage. But the naked eye is not sufficiently accurate for our purposes. A fluorimeter measurement of fluorescence present in each of the tissues above was used to generate the graph at right. Now the white bar is the control group, while the gray bar is the massage groups. From this graph you can see that massage at both ages does increase sarcolemma permeability relative to the control. This is most evident in the 10M group, that's the first gray bar, while the error bars for the 30C and 30M data make those 2 results effectively the same. As the researchers stated in their paper, membrane permeability, as measured by IgG infiltration into muscle fibers, was not altered due to massage or age, and no significant interaction was observed between age and massage, indicating that independent of the age of the animal, massage did not induce damage to the muscle fibers at the load used in this study. In the previous slide, we saw a positive control image in which IgG fluorescence could be observed inside injured muscle cells. It's worth noting that in this study, no muscle fibers from any of the experimental groups exhibited fluorescence intensities that were close to the ones in the injured muscle. This indicates that 30 minutes of massage applied to young and old muscles using a load of 4.5 newtons does not induce overt fiber injury after 24 hours. This data should look familiar from our previous discussion of density histograms. Since the fluorescence images in the preceding data were not sufficient to give us a clear visual sense of where the fluorescence IgG was located, the researchers tried a different analysis. A computer algorithm calculated the difference between the bright fluorescence between the various cells and the background fluorescence inside the cells. This resulted in a series of ratios that are graphed above. On the vertical axis are the number of fibers. On the horizontal axis is the fluorescence intensity of that fiber. In essence, this shows the number of muscle fibers that were brightly fluorescent versus the number that were not for each of the 4 sample types. You can see above that A, C, and D show very similar results, while only B, corresponding to the 10M or young massaged rat group, shows a bimodal response, that is, having 2 curves. This shows the Sarcolemma permeability in the 10M group, where extracellular IgG entered the muscle cells of young massaged Norway rats through porations in the sarcolemma. As mentioned previously, the researchers hypothesized that young tissues would respond differently to massage than old tissues, but their expectation was that old tissues would be more permeabilized by massage rather than less. Explaining this observation drove the researchers to look outside a simple analysis of IgG fluorescence to find an explanation for their data. That explanation turned out to be satellite cells, as we'll see in just a moment. Now recall from our discussion of key terms that satellite cells are the stem cells of skeletal muscle found between the basal lamina and the sarcolemma, and when Pax7 positive satellite cells are activated by injury, they rapidly divide, differentiate, and repair or remodel nearby injured muscle fibers. Since their original theory, that the sarcolemma of old rats would be more prone to injury and poration than young rats, turned out not to be the case, the researchers in this study wondered if satellite cells might be the reason why young massaged animals developed some amount of sarcolemma permeability, while the old massaged animals did not. To answer this question, the researchers did a second immunohistochemistry study in which they targeted Pax7 positive satellite cells with fluorescent IgG. In the fluorescence images above, you can see yellow spots where fluorescent satellite cells are. The cells were also counterstained with a reporter dye that becomes fluorescent only when it binds DNA, and thus allows scientists to look at the nuclei of cells where most DNA is found. And these are the blue-green structures. Now remember that skeletal muscle cells are polynucleate, meaning they have many nuclei per cell. As you look from the 10C to the 10M images, that's the first row, you can see the number of yellow satellite cells increases. The same is true when looking across the 30C and 30M images in the lower row. But note also how there are more satellite cells in the 30C than there are in the 10C, that's the first vertical column. Here we begin to understand why younger tissue developed some amount of sarcolemma permeability when older tissue did not. Age and massage are both independently associated with a higher number of Pax7 positive cells. Look at the graph at the right. The white or control bars are lower at both ages than the gray or massaged bars. This shows satellite cells increase in both ages of tissue as a result of massage. But at the same time, both white and gray bars are lower in young tissue than in old tissue. This explains the result that young massaged tissues experienced some increased sarcolemma permeability, while old massaged tissues did not. Satellite cells, much more prevalent in old massaged tissue, rapidly repaired any damage to the sarcolemma in old tissues. Now that we've talked through all the results of the research done in this paper, let's talk about the discussion. The discussion section is broken down into 2 types of text here. The text that I've made red above is an observation about the data by the researchers, while the text in bold is the conclusion they draw from that observation, given the methods, structures, and scientific reasoning involved. Let's go through them point by point. First, we observed no differences in membrane permeability between any of the groups, indicating that mechanoporation between all muscles was similar, independent of age and treatment. Therefore, massage as a mechanical stimulus was not injurious to muscles of young and aged rats. This is important because it means that the load of 4.5 newtons did not injure muscle cells of either aged rat. Do no harm is our first principle as LMTs, and it's good that this research backs that up. Second, there was a bimodal distribution of IgG abundance in young massaged animals only, not in aged. This is of significance for the following. The load used to massage the muscles in this study was determined to not only be optimal for enhancing muscle regrowth and remodeling following disuse atrophy in young rats of the same strain and age, but also as an immunomodulatory load in young rats. Here, the researchers discuss how massage of young, healthy tissue can promote growth and remodeling, as well as reducing immune responses and inflammation following exercise and mechanical load on tissue. This is significant for any LMTs watching this presentation who work with athletes, or those recovering from injuries that include some amount of atrophy. Athletes frequently exercise muscles to the point of micro-tearing, and this research shows massage has positive benefits for reducing inflammation and increasing muscle regrowth and remodeling. While many of us knew that, this is excellent scientific proof at a cellular level. Third, massage was associated with a higher number of Pax7 positive cells, indicating that mechanical loading and not mechanoporation was a sufficient stimulus to increase satellite cell number in skeletal muscle regardless of age. Here, the researchers note the increase in satellite cells following massage in both young and old subjects. This suggests a cellular mechanism for many of the benefits of massage that we as LMTs are already aware of, and demonstrates those benefits in both old and young tissues. This might be something to mention when someone tells you they are too old for massage to be helpful. Fourth, an increase in mechanoporation in young, coupled with an increase in satellite cell abundance, suggests that massage is inducing mild fluctuations in the sarcolemma and extracellular environment, which are different in aged muscles. The importance of these findings needs to be further investigated. Mild fluctuations, not micro-tearing, but still changes to permeability, were different for young tissues compared to old when massage was applied. More research into these differences is warranted based on these findings. Now, this may seem obvious, but I always like to point out that if your research doesn't raise even more questions than it answers, you did not ask an interesting enough question. So what conclusions did the researchers draw from all this work? Let's find out. Conclusions. The conclusions, like the abstract, are a highly distilled part of any research paper, summarizing the findings in the clearest and most high level of terms. Here, the take-home message is that massage is a common therapeutic technique, and we need to understand it better, not just in young healthy tissue, but also in tissues that have been perturbed in some way, including simply through age. With that knowledge, we can perform better therapies and optimally improve function for our clients. And again, reiterating their key findings, the researchers remind us that this mechanical load did not injure muscle tissue, but did alter muscle cell membrane permeability and increase satellite cell activity, with probable beneficial impacts for both young and old tissues. Their research may help elucidate how massage helps muscles recover from injury. Now that we've talked about the conclusions of the research, we're left with some questions, and that's a good thing, since research almost always leads us to a new vista, where each bend in the path brings new sights to our eyes. Here, I have just a few questions that occurred to me as a result of reading this research. First, can cyclic compressive loading, or CCL, be used to improve the consistency and reproducibility of technique, and thus results, when studying massage in human populations? One of the great challenges in massage therapy research is that every LMT applies force and pressure slightly differently. That is a benefit for clients, but the lack of consistency can make studying massage a difficult task for researchers. With the CCL machine or designs like it, that variability of operator is removed. Perhaps with a CCL or something like it, massage therapy research could be improved as a whole, yielding clearer results with more reliable and actionable findings, including in human populations. Second, if mechanoporation is a mechanism of muscle tissue injury, can the results of this research be used to inform our manual therapies? In the immunohistochemistry positive control we saw, extracellular IgG was fluorescing inside muscle cells, showing that the sarcolemma had been injured or at least highly permeabilized by mechanoporation. If this is a common mechanism of muscle injury, can what we learned in this research study improve our therapeutic knowledge when it comes to treating muscle injuries? The findings in this study show an immunomodulatory effect for massage, and we know an immune response is an early after effect of injury that leads to inflammation. Since we now know light massage leads to an increase in satellite cell activity and thus to repair or regrowth of muscle tissue, our therapeutic responses may be altered when presented with a client reporting muscle injury. Third, as post-load massage has been shown to be immunomodulatory in rats, is there a role for it as a therapy in autoimmune populations? Since massage is immunomodulatory, could massage have applications in the ever-widening range of clinical cases that have been determined to be autoimmune? This is certainly an area for case studies and eventual research. While speculative, the results of this research study present tantalizing clues to a link between massage and autoimmunity. Fourth, is there clinical human relevance for the increased amount of satellite cells in aged rat populations? While rats are a common model for all mammals, and humans in particular, we do not yet know if the results of this research translate directly to humans. While there are imaging technologies that can be ethically used on live humans without resorting to biopsies, these are far from common in massage therapy research. Yet even without direct evidence of increased satellite cells in human muscle tissue following massage, and increasing with age, might there be some therapeutic opportunities in the hypothesis that human skeletal muscle is like rat muscle in this respect? Only further research will answer that question. We have now reached the end of this presentation of the LMT Continuing Education Series presented by the Massage Scientist. Congratulations on making it through. These are not easy or simple concepts, and I hope you have learned a great deal about not just muscle cell biology, but also the process and relevance of research in massage therapy. While this video is free for all to view and learn from, LMTs who wish to receive their 1 hour of research continuing education credit in the state of Washington can submit payment at the address above. Please, please, please do not forget to include your name, email, and license number, as these are necessary for you to receive your certificate. I also have a short feedback form available, and I hope you will take a moment to share your thoughts with me. The survey is completely anonymous and is used only to improve the value of future presentations in this series. I'm also happy to answer any questions you might have, and I encourage you to email me. You are also welcome to follow this channel as I add more research videos. Thank you so much for your interest and support. Massage therapy research is something I'm passionate about, something I hope you now agree with me on, and I'm grateful to have the opportunity to share it with you. Thank you again, and I hope you'll check back for more content in the future. Stay curious.